Hello and welcome to this Nature Research custom media webcast titled Resolving the Fibrotic Niche, a Single-Cell RNA Sequencing Approach. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by ACD. We'll begin the webcast with presentations from Dr. Ross Dobie, Postdoctoral Researcher for the Centre of Inflammation Research at Queen's Medical Research Institute, University of Edinburgh, and Jyoti Sheldon, Associate Scientist for Applications for Advanced Cell Diagnostics. We'll then move on to a question and answer session. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them today. And now, over to Ross.
Hello, my name is Ross Dobie. I'm a postdoc at the Centre for Inflammation Research at the University of Edinburgh. I've got to say that it's great to have this platform to discuss my recent research in this presentation entitled Resolving the Fibrotic Niche, a Single-Cell RNA Sequencing Approach. For the past 5 years, while working in Professor Neil Henderson's lab, my research has been focused on understanding the cellular and molecular mechanisms that drive liver fibrosis, with an emphasis on mesenchymal cells. Now, before I dive in and start discussing our work, I first want to highlight the burden of chronic liver disease. It is estimated that a staggering 884 million people worldwide have chronic liver disease, with 2 million deaths per year. Put simply, liver disease is a huge problem and it's not going away. The graph on this slide shows the magnitude of the problem here in Scotland, shown in dark blue, where the death rate is higher compared to other Western European countries. Now, although there are numerous different causes or types of liver disease, some examples shown here, what they have in common is if left untreated, they result in scarring. Now, this is an important part of the normal wound healing process. However, if the underlying injury continues, this results in abnormal levels of scarring, termed fibrosis, and eventually leads to cirrhosis, where the level of scar tissue is such that it prevents the liver from carrying out its vital functions. It is well recognized that the degree of fibrosis predicts adverse outcomes in patients, and effective antifibrotic therapies to treat chronic liver disease are urgently required. Currently, there are no approved antifibrotic therapies, and often transplant is the only option. I've included these histological images from a review in radiographics as they nicely highlight the progression of fibrosis and the sheer extent of scar tissue in a cirrhotic liver and the complete loss of normal architecture. To enable the identification of new antifibrotic therapies, there first needs to be a strong understanding of the cellular and molecular mechanisms that drive the condition. To address this, our lab uses single-cell RNA sequencing to interrogate individual cell populations at unprecedented resolution. There is no doubt that single-cell transcriptomic technologies, such as single-cell RNA sequencing, are transforming our understanding of cellular diversity and function in both health and disease. They allow, amongst other things, the identification of novel cell types and subpopulations, the mapping of cell states and transitions, and the analysis of cellular interactions. In this presentation, I'm going to discuss 2 recent projects in our group which involved single-cell RNA sequencing. For the first part of the talk, I will discuss how we utilized this technique to uncover zonation of function in mesenchyme during liver fibrosis. This work was predominantly carried out on well-characterized fibrosis mouse models. I will then, in the latter half of the presentation, discuss our work looking at interrogating some of the mechanisms that regulate human liver fibrosis. As I'm sure many of you already know, the liver is a beautifully uniform structure composed of hexagonal lobules in a honeycomb-like pattern. At the periphery of these lobules, oxygen and nutrient-rich blood from the portal venules and the hepatic arterioles enters. The blood then flows through the small capillary network called sinusoids that run to the central vein, where the blood drains. The hepatocytes, which line up between the sinusoids along this portal central axis, show remarkable biochemical and physiological heterogeneity, known as metabolic zonation. Recently, two fantastic papers from the Iskovits group at the Weizmann Institute utilized the strengths of single-cell sequencing to obtain a zonation profile of hepatocytes and endothelia across the lobule. However, little is known about the zonation of mesenchyme. Hepatic stellate cells, or stellates, as I'll now refer to them as, are a type of mesenchymal cell. In the healthy liver, stellates, the green cells in this figure, reside in the space between the hepatocytes and the sinusoids, running along this portal central axis. We were keen to investigate whether, like hepatocytes and endothelia, do stellate gene expression patterns change relative to their position across the lobule. Following liver injury, these vitamin A storing cells have a crucial role in the pathogenesis of liver disease. They differentiate into myofibroblasts and represent the major source of pathogenic extracellular matrix deposition, and thus have attracted considerable interest as a potential antifibrotic therapeutic target. It's worth mentioning that there are other mesenchymal cells in the liver. Fibroblasts reside in the portal niche, providing structural support to the vasculature of the hepatic artery, the portal vein, and the bile duct, and have been shown to contribute to scarring in certain etiologies of liver disease. And vascular smooth muscle cells, or VSMCs, surround larger vessels, such as the hepatic artery. As we were initially interested in gathering information from all mesenchymal populations in the liver, it was important we selected the correct transgenic reporter and carried out the appropriate digestion protocols. Unfortunately, mesenchymal cell research is often hindered by the lack of robust positive markers that allow positive selection for FACS sorting. Furthermore, mesenchymal cells, and in particular stellates, to put it politely, are one of the trickier populations to isolate, and therefore require optimized digestion protocols. For this study, we used the PDGFRB BAC-EGFP reporter, which faithfully labels cells which are actively expressing the pan-mesenchymal cell marker, platelet-derived growth factor receptor beta. In the end, we actually settled on two digestion protocols and gating strategies for cell sorting. One was based on the 2015 Medirac Nature Methods Perfusion Protocol, and it was used to enrich for stellates. And another was a lab-optimized protocol enriched for fibroblasts and VSMCs. During this presentation, I will focus on the data from the samples processed through the 10x Genomics single-cell RNA sequencing workflow. We sequenced over 12,500 mesenchymal cells from 3 healthy mouse livers. Clustering of these cells revealed 3 distinct clusters, as shown in the TSNE. For those of you who are not overly familiar with the TSNE plot, this is a great way to plot multidimensional data in 2 dimensions. Each spot represents a cell, and cells with a similar gene expression profile cluster together. Identified marker genes, which are listed alongside each cluster, already give a hint to the cell type each cluster represents, and indeed immunofluorescent staining of selected markers, along with the pan-mesenchymal cell marker PDGFR beta, confirmed 3 topographically distinct populations. CD34, a marker for the orange cluster, labelled fibroblasts, which reside in the portal niche as shown by the yellow arrow in the top left image. Relin, a marker for the green cluster, beautifully labelled the hepatic stellate cells throughout the parenchyma, while calponin-1, a marker for the blue cluster, labelled vascular smooth muscle cells surrounding the hepatic artery and portal vein. As we interrogated the data, looking for the expression of our favourite genes and so on, what struck us was the broad expression of some classic nailed-on stellate markers across multiple populations. For example, Desmin, or DES, which has long been considered a specific stellate marker, was found to also be highly expressed in VSMCs. Although some differences may be due to the different techniques used to assess specificity, it's certainly food for thought. The other thing that caught our attention was the variation in expression of stellate marker NGFR, highlighted in the box. Clearly, the expression of NGFR was not uniform across all stellates. With this in mind, we isolated the stellate cluster from the other mesenchymal populations. Using independent component analysis, or ICA, we were able to identify a set of 81 highly variable genes in uninjured stellates. When we plotted the expression of these genes, as shown in the heat map, it was clear that we had two opposing signatures, 52 genes associated with one signature on the left-hand side of the heat map, and 29 genes associated with the other signature on the right-hand side of the heat map. Red equals high expression, blue equals low expression. Now, if you imagine all the stellates are lined up along the top of the heat map, on the left-hand side, we have cells that have high expression of genes including NGFR and ITGB3, and low to no expression of genes including ADAM-TSL2 and RSPO3. Conversely, if you now look at cells on the right-hand side of the heat map, they have low to no expression of NGFR and ITGB3 and high expression of ADAM-TSL2 and RSPO3. We were obviously keen to know if these opposing signatures related to topographically distinct populations in the liver. Indeed, for all single-cell experiments, validation is key. We used a combination of immunofluorescent staining and RNAscope, which allows spatial analysis of gene expression to help answer this question. Ideally, we wanted markers that were mesenchymal cell specific to make interpretation of the images easier, which did narrow down the list a little. RNAscope allowed topographical validation of markers for which no suitable antibodies were available, and the ability to use this in combination with antibody staining was crucial in these validation studies. NGFR and ADAM-TSL2 were identified as good markers for each stellate signature, as they were strong differentiators and they were also mesenchyme specific. Using well-recognized markers for hepatocyte zonation, we identified periportal, E-cadherin positive, and pericentral, CYP2E1 positive regions of the liver lobule. What we found is that the markers we identified for the opposing stellate signatures beautifully defined two topographically distinct populations across the parenchyma. NGFR positive stellates were found in the periportal, that is the E-cadherin positive regions, and ADAM-TSL2 positive stellates within the pericentral, that is the CYP2E1 positive regions. This led us to annotate these populations of stellates as central associated hepatic stellate cells and portal associated hepatic stellate cells. We, however, didn't just rely on the topography validation from these two markers, but confirmed this zonation using other markers identified from the ICA analysis. RSPO3 showed a similar expression profile to our central associated marker ADAM-TSL2, and, like ADAM-TSL2, it labeled central associated stellates, as indicated by the yellow arrows. Again, RNAscope allowed us to gain vital spatial information when no suitable antibodies were available. ITGB3, also known as integrin beta 3, showed a similar expression pattern to our portal-associated marker NGFR. And like NGFR, it labeled portal-associated stellates. Now, interestingly, both RSPO3 and ITGB3 have previously been identified as landmark genes used to zonally define endothelia across the liver lobule, which suggests potentially a possible spatial correlation between these two lineages. To quickly summarize our findings in healthy liver, our data shows that stellates divide into two topographically dimetric lobule regions, called portal-associated hepatic stellate cells and central-associated hepatic stellate cells. We were next keen to see what role, if any, this zonation played in fibrotic injury. Since the discovery around 35 years ago that stellates are the major collagen-producing cells in the liver, the potential to transition to this activated myofibroblast phenotype is thought to be equally distributed across the entire population. To induce fibrotic liver injury, we used the carbon tetrachloride CCl4 model, which is a robust model of central lobular fibrosis and recapitulates many of the features of human disease. Livers from mice administered CCL4 have what is called a bridging fibrosis, as is seen in these PSR images. Now, it's well recognized that stellates are the dominant contributor to this type of liver fibrosis. And in accordance with this, these immunofluorescent images show LHX2 positive, PDGFR beta positive stellate cells within the fibrotic niche of a fibrotic mouse liver. For our sequencing experiments, we again used the reporter strain described previously, as we were interested in collecting data from all mesenchymal populations. We pooled this data along with our uninjured dataset to give a full dataset of just over 23,000 cells. By and large, the initial clustering looked similar, with the three distinct populations representing stellates, fibroblasts, and VSMCs. Markers are listed alongside. However, when we split the data by condition, it was clear that there were specific regions, the two horizontal peaks, within the stellate cluster that were dominated by cells isolated from fibrotic liver. Interestingly, when we mapped our uninjured zonation signatures, that is the 81 genes, onto the stellate cluster, it was clear that these areas, the two horizontal peaks, were dominated by cells with a central associated stellate signature, shown here in brown. Now, to identify the myofibroblasts in the dataset and to investigate whether our zonation signature correlated with a myofibroblast phenotype, we created a myofibroblast signature by thresholding stellates based on their fibrillar collagen overexpression, that is COL1A1, COL1A2, and COL3A1. Now, this is a hallmark of activation. As predicted, stellates within the two peaks were classified as myofibroblasts, the green dots on the TSNAs. This signature was then mapped onto our zonation profile to identify the contribution of stellates from each zone to the myofibroblasts. In this way, we identified central associated stellates, the right-hand TSNA, as the dominant pathogenic collagen-producing cell, representing 88% of myofibroblasts classified in this manner. These findings were then again validated by spatial mapping in fibrotic liver, using a combination of RNAscope and immunofluorescent staining, which clearly showed ADAM-TSL2 central associated stellates located throughout the fibrotic niche, whereas NGFR portal associated stellates resided predominantly in the parenchyma, distal to the fibrotic region. Before moving on, at this point I'm going to quickly sidetrack to say that our full uninjured plus fibrotic dataset is available to peruse online. In this interactive browser, you're able to select your gene of interest and look at its expression across the full dataset and in response to fibrotic injury. I hope some of you will find it a useful tool for your research. In order to interrogate the dynamics of stellate differentiation to myofibroblasts in the different zones, we switched to a more dynamic model, an acute injury model, where a single dose of CCL4 is administered and the liver collected following 72 hours. At 72 hours, LHX2 positive PDGFR beta stellates have flooded the sites of injury and are producing extracellular matrix. There is also a high level of stellate proliferation at this time, measured here by EDU incorporation. Now, for this experiment, we focused just on stellates, sequencing over 7,000 cells from 3 mice following acute liver injury. Mapping of our zonation signature onto this data confirmed an obvious split within the stellate population. Portal associated stellates are coloured blue, central associated are coloured brown. In this acute setting, central associated stellates had a higher expression of fibrillar collagens, such as COL1A1 and other pro-fibrogenic genes, compared to portal-associated, indicating again that these cells are likely the dominant collagen-producing population. In this data, we also saw a strong proliferation signature, as indicated here by Ki67 expression, which seemed to be split between the two zones. Again, we validated these observations topographically. Here, we used immunostaining in combination with RNAscope, as well as dual RNAscope staining. This allowed us to assess our zonation markers in relation to cells which were actively expressing COL1A1, a major fibrillar collagen. These images confirm that cells which are actively expressing COL1A1, in green, are also positive for central associated stellate marker ADAM-TSL2, shown on the right, and not portal associated marker NGFR, shown on the left. With an interest in trying to investigate the dynamic process of stellate activation to the myofibroblast phenotype, we next informatically assessed cellular dynamics in the acute dataset. We first used the VelocCyto-R package to interrogate this injury response by calculating cellular velocity from unspliced, nascent, and spliced mature mRNA content. This can be exploited to estimate the rates of gene splicing, and in doing so, reveal the rate and direction of change within a single-cell dataset. The black arrows on the far left TSNE highlight the expected velocity within the cluster. What is clear is that the expected velocity is mostly restricted to the central associated population. In doing this analysis, we also found that the likelihood of transition between the two zones was negligible. Knowing that there was unlikely a switching between the two, we then used the monocle R package to independently define trajectories for both. While portal-associated stellates displayed only a proliferative response, illustrated by the orange arrow, central-associated stellates had a branching trajectory, with one branch displaying a primarily proliferative response, while the other branch represented the transition to a myofibroblast phenotype, illustrated by the green arrow. Taking this approach then allowed us to focus in on the specific changes in gene expression that exist along the identified central associated trajectories. This heat map shows the change in gene expression along the activation branch to the right and the proliferation branch to the left of the heat map. Red equals high expression, blue equals low expression. Again, imagine all the central associated stellates are lined up along the top of the heat map according to their identified place in the trajectories. Module 1 in green represents the genes that are upregulated along the activation branch, and Module 2 in orange, the genes that are upregulated along the proliferation branch. The spline curves show the relative expression of each group of genes, each module, across the trajectories, and the gene ontology terms confirm their classification. Although in this slide I've focused in on the known markers for activation and proliferation in order to define the modules, I think it is this sort of analysis that is crucial in helping us better understand the mechanisms governing cell transition. Here, it can be used to uncover new exciting mechanisms that drive stellate transition into myofibroblasts, and in doing so will be the first step in identifying new antifibrotic therapies. Having identified central associated stellates as the predominant pathogenic collagen-producing cell during murine liver fibrosis, we next wanted to look at ways of specifically targeting these cells. We identified that following acute and chronic liver injury, LPAR1 expression was restricted to the central associated stellate population. The LPAR1 gene encodes lysophosphatidic acid receptor 1, which is a G protein coupled receptor that binds the lipid signaling molecule lysophosphatidic acid, LPA. Previous studies have shown that LPAR1 expression is restricted to non-parenchymal cells and elevated in activated stellates, with minimal expression reported in other lineages, including hepatocytes. What particularly attracted us to LPAR1 was that at this point, we were also working on a human project, which I will go on to talk about more in the second half of the presentation. This meant that we had data from mesenchymal cells isolated from healthy and cirrhotic human liver. We had identified a cluster of mesenchymal cells which were enriched in cells from the cirrhotic liver, the blue cluster in the top left TSNE. These cells showed high expression of fibrillar collagens, as we would expect to see in myofibroblasts, and were also marked by LPAR1 expression, suggesting that LPAR1 was also a marker of myofibroblasts in cirrhotic human liver. Through an exciting collaboration with research-based biopharmaceutical company Gilead, we were also able to interrogate bulk sequencing data from human liver samples at different stages of fibrosis progression. We found that not only did LPAR1 expression increase in the later stages of disease, but the expression also correlated with expression of fibrillar collagens. To take this a step further and determine a functional role for the LPA-LPAR1 signaling pathway, again through our collaboration with Gilead, we were able to show in a rodent model of fibrosis that LPAR1 antagonism markedly reduced liver fibrosis as measured by both picrocereus red staining and alpha-SMA, as well as a hydroxyproline assay, with the ultimate aim of a therapeutic being to target the pathogenic population without perturbing the homeostatic role of other populations. This work highlights the strength of single-cell data to help identify new targeted therapies. To summarize this section, the data that I have presented deconvolves for the first time the entire hepatic mesenchyme in healthy and fibrotic mouse liver. We reveal spatial zonation of stellates across the hepatic lobule, partitioning them into two topographically dimetric lobule regions. Furthermore, we uncover functional zonation, identifying that central associated but not portal associated stellates are the dominant pathogenic collagen-producing cells in a mouse model of central lobular liver injury. Finally, we identify LPAR1 as a therapeutic target on collagen-producing HSCs in mouse and human, and demonstrate that pharmacological antagonism of LPAR1 inhibits liver fibrosis. For the second part of this presentation, I'm going to switch topics and now discuss our work focused on interrogating the mechanisms that regulate human liver fibrosis. As an introduction, I need to highlight that fibrosis is complicated. While you don't need to take in the details of this image, what I hope it gets across is that even the process of stellate activation is a complex one, and it's controlled. It involves numerous cell types working together and communicating with one another. Now, over the past 30 years, fantastic progress has been made to advance our understanding of the disease mechanisms underpinning fibrogenesis. But most of this work has been driven by work on rodent models. There remains a considerable translational gap between putative targets and effective patient therapies. Now, this is in part due to the limited understanding of the human condition, of the functional heterogeneity and the interactome of cells that contribute to the fibrotic niche in human liver disease. To try and address this problem, we completed a human-based study of cirrhotic liver. We collected samples from 5 healthy livers, taken from non-lesional liver tissue, obtained from patients who were undergoing surgical liver resection for a solitary colorectal metastasis, and 5 cirrhotic samples from patients undergoing liver transplantation. Much like the mouse studies, cells were isolated from these samples by enzymatic digestion and FACS sorted to enrich for non-parenchymal populations, and processed for single-cell RNA sequencing using the 10x pipeline. Now, this project made excellent use of the close proximity of the Scottish Liver Transplant Unit and our research institute. Because it was important that we try and minimize artifact, we developed a rapid tissue processing pipeline, obtaining fresh, non-ischemic liver tissue, which was taken before the interruption of vascular inflow during surgery, and then immediately processing this for FACS. This enabled a workflow time of under 3 hours from patient to single-cell droplet encapsulation. In total, we sequenced just over 66,000 cells from our 5 healthy and 5 cirrhotic livers, creating a huge dataset. Basic clustering of these cells confirmed isolation of all the major non-parenchymal cell populations across both healthy and cirrhotic liver. Much like the complex process of fibrogenesis, analysing this data was the work of many of the group working together. Because my main interests lie in mesenchymal cell biology, I will initially focus in on this population, the pink cluster in the middle of the TSNE. Now, looking specifically at the mesenchyme, our first aim was to identify the collagen-producing myofibroblasts. Isolating the mesenchyme from the full dataset and reclustering identified 4 clusters, as shown in this TSNE, with associated marker genes shown in the heat map. Clusters 3 and 4 immediately caught our attention, as they were clearly heavily represented by mesenchyme from cirrhotic liver. Marker genes such as KRT19 and MSLN allowed us to confidently label cluster 4 as mesothelial cells, which are found on the surface of the liver. Cluster 3 showed increased expression of numerous genes associated with myofibroblasts, such as the fibrillar collagens. To spatially map these populations, we took a similar approach to the mouse work. Immunofluorescent staining of selected markers was used to topographically identify the populations in healthy and cirrhotic human liver. Co-staining with markers for each cluster, MyH11, a marker for cluster 1, RGS5, a marker for cluster 2, and PDGFR-alpha, a marker for cluster 3, confirmed the presence of 3 topographically distinct populations. MyH11 is a classic VSMC marker, and unsurprisingly, it lit up VSMCs in the liver. RGS5 staining was observed in a population of cells in the parenchyma in healthy and cirrhotic liver, consistent with stellate cells. PDGFR-alpha staining was minimal in healthy liver, but clearly labeled a population of mesenchymal cells in the fibrotic niche. Using QPath software to segment the cirrhotic images into nodules and fibrotic septae, we were able to confirm that this staining was localized to the fibrotic regions. Taken together, this information allowed us to confidently annotate cluster 3 as our myofibroblasts. A similar approach to the one I have described above was taken for characterizing the other non-parenchymal cell populations in healthy and cirrhotic liver. Here I have focused on the macrophages. I will not go into too much detail, but to summarize, we identified a population of monocyte-derived, CD9 positive, TREM2 positive, scar-associated macrophages in the cirrhotic liver, which displayed a pro-fibrogenic phenotype. Immunofluorescent co-staining, shown on the left, and RNAscope, again combined with immunofluorescent, shown on the right, confirmed the presence of these CD9 positive, TREM2 positive macrophages within the fibrotic niche. MNDA was used as a marker of macrophages. Antibodies for these marker genes also allowed the successful isolation via cell sorting of the scar-associated macrophages from human cirrhotic liver. Previous studies in rodents have highlighted that macrophage subpopulations orchestrate both the progression and regression of liver fibrosis. When added to primary human stellate cells, conditioned media from isolated CD9 positive, TREM2 positive macrophages promoted fibrillar collagen expression, indicating that these cells have a pro-fibrogenic phenotype, similar to the one observed in rodent models. The conditioned media also increased proliferation of primary hepatic stellate cells. Having identified populations of scar-associated macrophages and mesenchymal cells, or myofibroblasts, our interests turned to working out the ligand-receptor interactions between these populations. By working out these interactions that exist between cell types, we hope to uncover highly relevant intrascar pathways that could be potentially druggable. To assess these interactions, we used CellPhoneDB, an amazing resource created by the Teichmann Lab in Cambridge. CellPhone allows the prediction of enriched cellular interactions between 2 cell types from single-cell transcriptomic data. For the purposes of this presentation, I will focus in on the macrophages, labeled as SAM, and the myofibroblasts, labeled as SAM-ES interactions. Within the CellPhone output shown in this slide, circle size indicates p-value and color denotes average ligand and receptor expression levels in the interacting populations. Numerous significant interactions were detected between ligands and receptors expressed by these populations. An example is the ligand PDGFB and its cognate receptor PDGFR-alpha. Now, PDGFR-alpha was, of course, already identified as a marker of the myofibroblast population for topographical validation. PDGFB was found on TREM2 positive macrophages within the fibrotic niche, as indicated by the white arrows in the immunofluorescent image. To assess the functional role of this interaction, we turned to cell culture studies. PDGFB induced proliferation of primary human hepatic stellate cells, which was inhibited by blockade of PDGFR alpha, the receptor. Taken together with the previous data showing that cultured media from scar-associated macrophages promoted hepatic stellate cell proliferation, this data points to a functional role for scar-associated macrophages in regulating myofibroblast cell expansion through the PDGFR alpha signaling pathway. As mentioned before, this is an example of the many interactions identified in our dataset, each with the potential of being a tractable target to inhibit human liver fibrosis. In summary, the data that I have presented in the second part of this presentation gives an insight into how single-cell RNA sequencing and spatial mapping was used to resolve the fibrotic niche of human liver cirrhosis, leading to the identification of novel pathogenic subpopulations of TREM2 positive, CD9 positive, fibrogenic macrophages, and PDGFR alpha positive collagen-producing myofibroblasts. With this increased resolution, we were able to dissect the complex pro-fibrotic interactome between these novel scar-associated cells and identify highly relevant intrascar pathways that are potentially druggable. Now, all the work that I've presented would not have been possible without the contribution of so many people, and I apologize if I've missed anyone out. In particular, I would like to acknowledge John Wilson Kanamori, who was the informatics lead on both these projects, and Prakash Ramachandran and his group for leading what became a very large and fascinating human-based liver project. Finally, I would like to thank Advanced Cell Diagnostics for inviting me to discuss our research. It was a great opportunity and I've thoroughly enjoyed it. Thank you.
Thank you so much, Ross, for that amazing presentation. Now I'd like to focus on the applications of RNAscope in various areas such as liver research, as well as how you can confirm and spatially map diverse gene signatures by incorporating our multiplex in situ hybridization assay in single-cell RNA sequencing workflows. My name is Jyoti Sheldon and I work in the applications team at Advanced Cell Diagnostics. So let's look into the technology of RNAscope. What is RNAscope in situ hybridization technology? The RNAscope technology is an ideal spatial analysis solution to interrogate complex tissues. It is both highly specific as well as a sensitive method to detect RNA biomarkers in cells and tissues while giving you that morphological context. But then you can do all of this at a single cell resolution or a single molecule resolution. Now, the technology itself consists of three parts. On the left side, you see the first part is a unique target probe that ACD will design against your sequence of interest. Then the second part is a signal amplification system, which is our patented amplification system that designs or generates a high signal to noise ratio. And lastly, you'll see the detection and quantification, where you can visualize your data as a single RNA molecule or dot. Now, this assay allows you to spatially map various kinds of RNAs, such as mRNA, long non-coding RNAs, splice variants, and specifically highly homologous sequences. Just due to our high specificity, you can discern between sequences that have high homology in both cells as well as intact tissues. And all of this can be visualized using either a fluorescent or a chromogenic label. Now, this assay can be performed on a wide variety of sample types. Some of those are the FFPE tissues, both the fresh frozen as well as the fixed frozen tissues, PBMCs, and cultured cells. So how does the technology work? The two key features of the RNAscope technology are the probe design and the signal amplification. The oligonucleotide target-specific probes, they're depicted as these two Zs to emphasize the fact that they're linked by a spacer. Now you see the bottom of this Z complements and hybridizes to your target sequence or the transcript, while the top of the Z creates a base for the amplification structure. So what happens when these two Z hybridize in tandem to the target sequence? It creates this binding site on which this preamplifier can then be built on and bind, and then the amplification tree can be built from there on. After the Z pairs hybridize with the target RNA, the pre-amplifier binds to the top of the ZZ pair, after which the amplifier will bind, multiple amplifiers will bind on top of the pre-amplifier. And then each of these amplifiers can further bind multiple labeled probes. The labeled probes contain either a chromogenic enzyme or a fluorophore that generates a signal detectable under a standard bright field microscope or a fluorescent microscope. Now, this particular signal amplification strategy yields a high sensitivity and allows you to visualize target RNAs as a single dot, where each of these dots represents an individual RNA molecule that can be quantified. Background, on the other hand, is completely eliminated because the signal is dependent on both these Z's binding next to each other on the target. What that means is you need both these Z's right next to each other on the target sequence for this tree to be built. If both these Z's do not bind, then the preamplifier cannot form a stable hybridization, and this amplification tree system does not get built. Consequently, no amplification of non-specific hybridization occurs, generating little to no background signal. A standard RNAscope probe that you will get will consist of 20 Z pairs pulled together that are designed to hybridize next to each other along a target region, allowing for a tremendous amount of amplification as well as signal potential. However, only a minimum of 3 Z pairs is needed to bind to the target RNA sequence in order to generate signal for molecular detection, which is really important in samples such as old archival FFPE samples with degraded RNA. Taken together, this combination of this probe design as well as the signal amplification ensures a high signal-to-noise ratio. So what are some of the RNAscope on BaseScope product portfolios or offerings? This slide summarizes all the assays that are currently available from ACD. We have both chromogenic assays as well as fluorescent assays. The chromogenic assays are available as singleplex brown or red or duplex assays that can be performed on both manual as well as automated platforms such as the Leica and the Ventana platform. The multiplex fluorescent assays allow up to 4 targets simultaneous detection in a single tissue section. The 2 fluorescent assays are the multiplex fluorescent and the multiplex fluorescent V2 assay. The difference is that the V2 assays uses the TSA from Perkin-Elmer for signal visualization and provides a high signal-to-noise ratio. Last year, we introduced our high-plex assay that allows for simultaneous detection of up to 12 targets on a single tissue section. If you are looking for targets that are short sequences and shorter than 30 base pairs, we have our BaseScope assays that are also available as BaseScope singleplex or duplex, and only the BaseScope singleplex can be performed on the automated Leica and Ventana platforms. In addition to all these assays, we recently launched our microRNAscope assay. The microRNAscope assay allows you to detect shorter targets such as ASOs, microRNAs, siRNAs, and RNAs smaller than in the range of 70 to 50 nucleotides. If you would like more information on this particular assay, you can reach to your local sales rep or the ACD support team, and they will give you more information on that. Now, let's go into how customers are incorporating RNAscope into their single-cell sequencing workflows. As you all are familiar that the current single-cell transcriptomic studies, they utilize dissociated cell, which results in the loss of spatial organization of the cell population in a tissue context. Hence, validation and spatial mapping of single-cell analyses can be obtained using assays that retain the spatial organization, such as the RNAscope in situ hybridization assay. Our multiplexing capabilities, coupled with the high signal-to-noise ratio, enable single-cell and single-molecule detection of transcripts in the tissue context, providing you with that confirmation of high-throughput single-cell transcriptomic results, as well as adding that spatial information to these data sets. Some of the common applications we've seen in literature for RNAscope technology in this single-cell sequencing workflows are to spatially map a cell atlas. You can also identify new cell types, classify highly heterogeneous cell types, confirm some of the new therapeutic cell types, characterizing immune landscapes, as well as identifying new immunotherapy targets, analyzing or predicting response to drug treatments, And lastly, confirming some of the currently publicly available data sets, examples such as the TCGA or the Tabula Muris. So where is RNAscope technology used for liver research? Some of the common areas we see in RNAscope technology applied in liver research is for studies of fibrosis and steatosis, for looking at liver zonation, in which spatial information is really important, to identify secreted proteins, for validating single cell data, as I talked about earlier, for confirming gene therapy editing events, in studies for hepatitis and CMV, as well as in hepatocyte development and renewal studies. So I've picked up a few publications to show you some examples of these applications, and then just go a little bit into some of our newer workflows that help you support a lot of these applications. Shortage of approved antifibrotic therapies exist, despite that researchers have a better understanding of the pathogenesis of liver fibrosis. In this particular paper from Queen's Medical Institute, as well as GSK, researcher McBride and his fellow scientists, they identified a small molecule agonist for relaxin, a GPCR RXFP1, for treatment of liver fibrosis. Now, they used RNAscope chromogenic assay to assess the expression of this GPCR target and visualize its distribution in human liver biopsy tissues from two types of liver fibrosis patients. What they observed was there was a direct association of the disease progression with the expression of this particular GPCR RXFP1, which really shows the significance of performing spatial analysis techniques. This particular paper is from Erasmus Medical Center that studies the pro-inflammatory cytokine response in liver during severe influenza. Pro-inflammatory cytokines can be detected in the lungs and blood of infected patients, but the role of extra-respiratory organs in the production of these cytokines is unknown. In this particular study, they used ferrets as their gold standard for studying human influenza. This is a really important paper highlighting our probe design strategy. ACD can make probes for any target and any species as long as you know your sequence of interest. So especially in cases such as outbreaks of viruses where you need targets or probes designed faster, ACD can design the probe for you within 2 weeks, where antibody development may take a longer time. They use the RNAscope-ish assay to identify the cellular source of several cytokines such as IL-6, IL-8, and TNF-alpha in extra-respiratory tissues from ferrets inoculated with this highly pathogenic avian influenza H5N1 virus, and showing you the presence of in this particular image, IL-8, but the paper also shows IL-6 and TNF-alpha. Now let's shift gears to look at an application of RNAscope in single-cell sequencing workflows for liver research. Two main epithelial cell types playing important roles in regeneration are hepatocytes and biliary epithelial cells, or BECs. This particular paper from Harvard University performed single-cell RNA sequencing to examine hepatocyte and BEC heterogeneity during mouse liver homeostasis and after injury. They uncovered that BEC heterogeneity is defined by active YAP signaling, which becomes induced in response to injury or bile acids. They used the RNAscope multiplex fluorescent assay in combination with immunofluorescence, adopting one of our new dual RNAscope-ish IF technique to reveal the BEC transcriptional heterogeneity as identified by single-cell RNA sequencing. The dual-ish IF technique allows you to simultaneously detect both RNA and protein on the same tissue context. I'd like to go over a little bit on the RNAscope Hiplex assay that was launched last year. As I mentioned before, the RNAscope Hiplex assay allows you to look at simultaneous detection of 12 mRNA targets on a single tissue section. The way this assay works is it consists of 3 different rounds, and each round you visualize 4 targets. So 4 times 3, 12. At the end of the 3 rounds, you register all your images using our image registration software that enables a true 12-plex data visualization at a single cell resolution. The fluorophore cleaving procedure between these three rounds has minimal to no effect on the RNA and tissue morphology. Currently, this assay is compatible with fresh frozen and fixed frozen sample types. So now I'm showing you an example of a paper that we used to show the application of this RNAscope HiPlex assay. In this paper by Goksha, published in Cell Reports in 2016, researchers at Stanford University identified novel subtypes of D1 and D2 striatal medium spiny neurons, or MSNs, by analyzing over 1,200 different striatal cells using single-cell RNA sequencing. They identified discrete cell types that exist in a continuous spectrum of transcriptional states and found that functional diversity within a complex tissue arises from a small number of discrete cell types. However, they did not confirm their single-cell RNA sequencing results, nor did they show the spatial relationship between the D1 and D2 subtypes. So we employed the RNAscope HiPlex technology to confirm these results and spatially map the subtypes in the mouse brain. This slide shows data from our RNAscope HiPlex 12 assay, where we compile the images using the HiPlex image registration software. With this assay, we could visualize the heterogeneity of these cell populations on the same tissue section. This looping image was created using online tools to more clearly visualize and highlight the data from this experiment. By doing so, you can clearly see which cells are expressing the various combinations of gene targets. This is a still image from the previous video, which shows simultaneous detection of both the D1 and the D2 striatal MSNs. But you really look at this image and you're like, there are so many dots. How do I understand this, right? How do I kind of deconvolute this data? And that's where the HiPlex image registration, this information today and all these publications, I really do want to highlight that the growth and adoption of this technology is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then we have over 2,700 peer-reviewed papers published using this technology in numerous journals, some of them in many top-tier ones such as the Nature, Cell, and PNAS, etc. The RNAscope technology is highly relevant across multiple fields of research. Our top areas are neuroscience, cancer, infectious diseases, and furthermore, the incorporation of this technology in single-cell RNA sequencing workflows is also demonstrated by a rapid growth of publications using this assay to confirm and spatially map single-cell RNA sequencing data. With all of that, I hope I've convinced you today that RNAscope uses a unique probe design strategy to dramatically improve signal-to-noise ratio. We have currently over 22,000 probes on catalog, but new probes can be designed within 2 weeks for any target and any species, as long as you know your sequence. RNAscope provides both quantitative and spatial information on your targets in the complex tissue environment at a single cell resolution. Our BaseScope assay can detect splicing variants and short sequences down to 50 nucleotides, and RNAscope can be complemented by IHC or IF for simultaneous RNA and protein detection on the same slide. With all of this information today, if you have any questions for us or if you would like more information, you can visit our website at www.acdbio.com, or you can contact us at the email addresses on the screen. Thank you so much for your time and attending this webinar, and I hope you're all staying safe and staying healthy. Thank you.
Thank you very much, Ross and Jyoti, for those presentations. It's now time for our question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. So our first question, Ross, what challenges did you face combining RNAscope with immunofluorescence?
I think one of the most important things to do when combining RNAscope with immunofluorescence staining is to optimize your immunofluorescence antibody with the pre-treatments required for RNAscope. In our work, antibody staining was always completed following the RNAscope protocol
just because an antibody works with a standard immunofluorescence protocol does not necessarily mean it will work when combined with the pre-treatment required for RNAscope. It is definitely worth taking time to optimize your antibody staining with your selected RNAscope pre-treatment timings before combining the two protocols.
Okay, thank you very much. And were there any other additional challenges that you faced when carrying out RNAscope on diseased fibrotic tissue?
What additional challenges did we face? Autofluorescence was a challenge when dealing with fibrotic mouse liver. We would standardly use size 3 or fluorescein pyramids to visualize our RNAscope. But because of the high levels of autofluorescence observed in these channels, we had to switch to different pyramids for detection in fibrotic tissue, such as size 5. It's something that's probably worth considering before starting an experiment.
Okay, thank you very much. Our next question, how is it possible to identify secreted proteins with RNAscope?
Hi, thanks for the question. So we're not identifying the protein. Of course, RNAscope is for detection of RNA, but it's an application in secreted proteins because oftentimes the antibodies for these proteins are difficult to get and can be very non-specific. So RNAscope provides as a great alternative to look at some of these targets on a tissue context.
Thank you very much, Jyoti. And another question coming in for you. What level of plexing can be done with the RNAscope assay?
That's a good question. So at the moment, we have an assay, the high-plex assay, as I mentioned in the presentation, that allows to simultaneously detect up to 12 targets. But we are launching a version of that assay that will allow users to expand their targets to 48 simultaneous target using our newer reagents. So you will be able to visualize 48 RNA targets on the same tissue section by doing multiple rounds. Thanks.
Thank you very much. A further question, when designing short probes to avoid cross-reactivity with related family members, how much signal loss can one expect compared to longer target sequences? Jyoti.
Sure. So based on our probe design strategy that I mentioned earlier, our amplification tree that is being built on, and the way the probes are designed using the algorithm, you should not have any loss of signals when comparing longer targets to shorter targets. And our probe design team, they measure the feasibility of these probes when they design it for you and make sure that the signal is retained regardless of the size that you are trying to detect. But if you have any additional questions on like high homology targets, you can obviously reach out to our support team and then they can work on those things case by case basis.
Thank you very much, Jyoti. Our next question, I think, is for Ross. How best can we isolate mesenchymal cells from, I think that's virgin tissue?
And so just talking from our experience of isolating mesenchymal cells from the liver and various other tissues, I would suggest using the pronase digestion enzyme. It certainly helped us when isolating great numbers of cells.
Thank you, Ross. Would you like to add anything, Jyoti, on that?
Sorry, I was on mute. I think that was a question for Ross. Yeah. No.
Okay, I will move on then. Our next question is for Ross. What is next for the project?
So if we just focus on the human work, what you'll have noticed from the presentation is that we were dealing with cirrhotic liver, where the fibrosis has likely established over many years and could be considered more end stage. And what we're now interested in is carrying out similar work, but during the evolution of fibrotic liver disease, finding out how the pathogenic cells interact during the different stages of disease evolution and fibrosis progression, and how these interactions change, I think will be very interesting.
Definitely. Thank you very much, Ross. And for you, Jyoti, can the multiplexing targets be combined with an antibody?
Yeah, sure. So as Ross showed in his data, we do allow the RNAscope actually does allow you to combine RNA with antibody. That being said, the technique that Ross used, as he mentioned, where he first ran the RNAscope target followed by detection of the antibody, has recently been improved, and we are launching our new workflow called the co-detection workflow that enables researchers to apply any antibody to their RNAscope panel while preserving that antibody signal. So more information on this can be seen on the website. We also have a webinar tomorrow for the launch of this new workflow if you want to learn more. But any antibody can be combined with either our singleplex, duplex, or our multiplex workflows and assays. Thank you, Sarah.
Thank you very much. And that is where we need to finish for today. I'd like to thank Dr. Ross Dobie and Jyoti Sheldon once again for the presentations and for answering your questions. I would also like to thank the webcast sponsor, Advanced Cell Diagnostics, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.